Hello, everyone. Uh, Move out of the way of my own background. Hi, I'm John Quinn, Director of Science and Product Development. There is my email address, john.flojo@bd.com. And today we're doing the Data Wrangling Throwdown, where we talk about all things that lead to feeding data into a clustering or a dimensionality reduction algorithm and really deep dive on it. And we will speak nothing at all about the actual clustering, nothing at all about the dimensional reduction, but really focus on the steps that end up helping you to produce good outcomes upstream of doing any of that kind of work. So happy to have you here for our last webinar of the year. Happy 2024, going into 25 for everyone. And I think let's do this. The format that I'm going to go with today is the text is first. The text is reversed. For me, it's the right way. And I actually automatically, I reversed it so that it would look right. Yeah, oh well. It's just a background. We'll move past it in about 3 seconds. The format for the rest of the time is that we are going to, I'm going to bring up a few PowerPoint slides to structure the discussion and intermittently weave in the how to do it in Flojo as I go through the various topics that we're going to talk about. So the text is backwards. It's just john.flojo@bd.com. I am in large part involved in helping figure out what should go into the software. So if you have any great ideas or read any papers, something like that, happy to hear about it. Also happy to hear about complaints and tech support and so forth. And you can also write flojo@bd.com for our general tech support. All right, so with that, why don't I share my screen and get going with some PowerPoint? Okay. And we can close that, we can allow that, and go full screen. Okay, so there you go, a not backwards version of my email address then. For all of, and you know what I meant to say, and I meant to do as well, that I'm going to make sure that I've got the chat open the entire time. There we go. Let me make sure that is open. And so this is true. The chat open the entire time in Zoom, so that if you've got questions, just type them into the chat. We will attempt to go through them right away. Thank you also to those who sent in emailed questions in advance. I copied and pasted them right into the slides. So we will make sure that we address your questions that you sent in advance. Okay, so webinar outline. Let's talk a little bit about the steps that lead into doing good clustering, good dimensionality reduction, and so forth. So quality control, metadata management, normalization and scaling, concatenation, and then after that, you do great things. We'll talk for a moment about the purpose of each one of these steps, review super briefly the options. There are lots of webinars out there that get into the nitty-gritty of each one of these components individually. I will probably tread very lightly on how every single algorithm works so that we can do a whole bunch of different things within the hour that we have together. Feel free to ask questions when there's something that's pressing, but just know that I'm not going to probably spend a whole lot of time on the details of each algorithm. And then I'll show how it works in Flojo, and then I will make sure I check the chat each time to see if there are questions, because we want to make this as interactive as possible. So, quality control. Okay, purpose of quality control. There are a lot of ways to ensure that you have gotten rid of technical anomalies for acquiring the data. And the purpose of it is to make sure things like the purple population don't happen to you. This is a slide that I use in a lot of different places just to show like a why is QC necessary. The idea here is this data set shown with time on the x-axis. Same plot here, just one with pseudo color and one with like a contour plot. You can see I've done gates to divide, the first 15 seconds or so up into this purple, the next into the gray, and then the next into the light blue. You can see that we've got really dense event packing through here and also some visible striation, as there was probably some micro clogs and misalignments happening, an actual clog, maybe that breaks out, and then a probably turning down the flow rate, etc. So all sorts of differences in flow rate. And then you can see that by color coding, when we start looking at all the other parameters, the purple population really only exists in those first few seconds when there was a lot of coincidence happening. And so it's probably not a real population. And that is what we want to avoid, having some kind of just artifact that happened during acquisition impact what you're going to conclude about your data. So what can we do to avoid this? Step one. There are a ton of data QC algorithms that are out there. It is a popular line of research because everyone who does bioinformatics has noticed that they need cleaner data to get better results. Here is a sampling of 4 that we have up on the Flojo Exchange. Flojo Exchange, just part of www.flojo.com, that is essentially our community outreach, our place where we can work together with anybody doing cool bioinformatics work and bring their work into Flojo so that you can just call it and run it within Flojo without needing to be a programmer. All those things are free. So if you have Flojo, you have the ability to access any of these algorithms. The 4 that I've posted here are Flow Clean, Flow AI, Peacock, and Flow Cut. You can tell going left to right based on the iconography how new they are and how much we were getting into just using various AI to make cool pictures and so forth. So I like basically all of them, and all of them are better than using nothing or trying to manually do any kind of QC. I will point out the 2 in the middle, Flow AI and Peacock, are my favorites, mostly because of the in the list down here of what they do. They both produce great reports, and I really like getting some kind of graphical output that tells me why anything was removed from my data, as opposed to trusting the algorithm blindly. They also do a full suite of signal checks that are nice: flow rate, signal stability, dynamic range, and so forth. So these are cool. You can use them. Before we actually, I'll demo using one. I'll mention double discrimination, another part generally of the QC. There's a variety of ways to do it. The way that I have illustrated here on the screen is just using a couple parameters. So forward scatter area versus forward scatter height. And the reason that I'm using those 2 parameters to try to remove doublets is that you can imagine there is actually a time element to a cell passing through a laser, illustrated in this picture. Whereas the cell enters the laser, you get some signal. It's maximized in the center of the laser, you get the maximum signal, and then as it passes through, it falls off. So you get this pulse, and we have to turn that pulse into a single number that says this is the intensity of this cell for a given parameter, so such as forward scatter. So you can integrate that pulse to get the area. That's the forward scatter dash A. You can take the height, forward scatter dash H. You can take the width, forward scatter dash W. And for a cell passing through the laser, things like the area of that pulse and the height of the pulse should be strongly correlated. And when they're not, that's probably because 2 cells were stuck together, or some debris was stuck to your cells, or anyway, generally unreliable signal was detected during that pulse. So I think that's a reasonable way to do it. It doesn't have to be done specifically with those 2 parameters, but that's the way I like to do it. If you've watched any of our last 2 webinars, which were all about using imaging technology, you saw that imaging technology is a great add-on to be able to identify single cells, but anybody can do this one who's got a cytometer. Okay, and getting rid of the dead cells. So getting rid of doublets and dead cells have the same general purpose, in that 2 cells stuck together could present, if you thought it was 1 cell, a really interesting and unique profile. That probably confounds your results. Dead cells are notoriously sticky and will bind everything. You'd like to remove them. The typical plan here is to do some kind of live dead dye, something that permeates the membrane of damaged dying cells and doesn't if the cells are healthy, and at least in large enough quantity to have them appear beyond background. Just showing here that you can also do a backgate assessment if you want to double check any of the gating that you've done in a manual manner. The only 2, really gates that I recommend doing: some kind of time-based cleanup, not really gate, and some kind of live dead, and some kind of doublet discrimination. And I advertised when I put up the ad for this webinar that we were going to talk some mass cytometry to make sure that we include the mass cytometry folks. So here's our interruption for some mass cytometry talk, because a lot of what I just said maybe doesn't particularly pertain to mass cytometry. And so the things that you can do in the pre-processing steps or data quality steps for a mass cytometry experiment is use an event length versus debris gate. So event length is essentially how many scans that the mass cytometer used, and generally more scans means it was a bigger wholer thing. It's more likely to be a whole cell, and so sort of the large population visible for DNA and event length are good to do. And then you could do something like cisplatin, which is what mass cytometry will use to discriminate live dead, for the case where you're maybe going to obliterate the cell at some point. I think there was a paper from the Nolan lab that came out a little whiles ago that advertised this. I will also point out that this is a more recent paper where this group here give you a whole bunch, you can see right here in the paper, of different markers that you can use in an attempt to identify viable cells in a mass cytometry experiment. And then the last thing is that amongst those QC algorithms, I'll point out that Peacock actually has a CyTOF setting. So not really a traditional flow rate in mass cytometry. Some of those time step parameters might be absent in your mass cytometry data file. CyTOF has got its own setting in Peacock that will work with that and allow you to do the QC using that particular tool. So there you go. And there's the paper again. I guess I turned it off. Okay, so before we move on to metadata management, let us move into FlowJo. Okay, and I've got many workspaces open, but that's the winner. Okay, so I've got just some data here uploaded into Workspace. I want to show you how to run one of the QC algorithms. I will skip drawing a few gates. I'm assuming that's okay at this point, and we will try to do that to save time. But if I wanted to run a QC algorithm, and this is actually mass cytometry data, actually from the Bendil paper where XShift was used to, make the sort of famous, if you've read it, the famous plot of dividing up cells by phenotype using mass cytometry data. I can go to the plugins drop-down menu here. The first item is the FlowJo Exchange. If you click that, that would take you to the website, the FlowJo Exchange, that has the place where you can get any one of these algorithms. Each one of these cards is an algorithm. So I'm going to use Peacock. If I've downloaded the algorithm and installed it, lots of videos available on that. And not too terrible. Basically, I put it somewhere on my computer. I guess I'll show you. I put it in a folder, my applications folder, and just say under diagnostics, the plugins are here. And at that point, anything in that folder loads into this list. Some of them depend on R, which would then require a step or two of copy pasting the actual R libraries to be loaded into your R. You'd have to have R, do that. But if you've got admin rights to your computer, that's not bad. If you don't, I have to work with IT. Good luck. Okay, moving on to running Peacock. I click it. I have a list of all the parameters I want to make sure I consider. Generally, you're going to pick all of them. Yeah, all of them. Boom, 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 boom. Okay, we can stop there. Check the CyTOF button if it's CyTOF. Uncheck the remove margins button if it is spectral data that you're working on and you're in Peacock. With many extra detectors, the remove margins button, which tries to remove any event that's stuck to either the upper or lower bound of your data set, will be removed. And it probably is overzealous if you're in spectral data with all those extra detectors. There is likely a detector that your cell did not express anything in, and it might be removed extraneously. Turn that off if it's spectral data. You've got a couple tunables here. What the Peacock algorithm will do is use an isolation tree and a median absolute deviation threshold to decide whether a cell is something that was a technical variant. Details are plentiful elsewhere on what those two things exactly are and what they mean. But end of the short story here is that the entire paper about using Peacock explains in great detail how the folks who wrote this, also listed here, came out of the Sayes Van Gassen team, done by Manuel. They derive those numbers empirically, and they work pretty well. And so using the defaults is generally just fine. If you want to save the R script for diagnostic purposes, you can. If you want the images to be added into your workspace, you can check those. Otherwise, hit OK. And it'll run for like 20 seconds or so. And while that's running, I'll show it. I ran it just before this started another file. What pops out at the end is the good events and the bad events. Those are exactly what they sound like they are. You double click on the good events and start doing your work thereafter. You may want to remove the Peacock node if you want. So there goes, that one's just finished up. You could drag it to the group to apply to all the files in a group individually. It'll just run on each one of those files. You could delete it afterwards, as most of our plugins are designed to rerun if you modify something. Good for a clustering algorithm, where maybe you're changing the input of which cell should be in this cluster. Not so good for data QC, where that's not really going to change. Anyway, so you've got that. If you well, if you chose or didn't choose to add the data to the layout editor, cool, but you also have a folder of the same name as the workspace, wherever you save the workspace, that if you want the graphics, they're in these Peacock results, Peacock plots. It's as a PNG file, and then you can see why those like 2% of the events were taken out. Look like we had a really tiny blip here in some parameter, and you can see it just shown consistently across where we did not have a stable signal, and it was taken out due to median absolute deviation as opposed to the isolation tree, and that removed the 2% of the cells that came out or so here. Okay, so that is running Peacock. Let's see, what do I have? I've got oh, people are typing in the Q&A. Sorry, I was not looking at the Q&A, which seems to make sense. Let's see, Peacock recommends working on transform data, doesn't it? The answer to that is yes. Good question. The rest of those QC algorithms would suggest that you run it on the uncompensated data. Compensation is a transform. Peacock will use a clustering step as part of doing the isolation tree. Again, for full details, watch a webinar on Peacock, which we have up there. It basically just means that it's a clustering algorithm that tries to identify the most easily separated cells. And because it's clustering the data upstream, working with the transform / compensated parameters is a good thing to do to get the best possible separation. Peacock margins are based on the range defined in the header of the SCF file. Yes. Is it best practice to run the cleanup algorithm on ungated data or after single slice gating? Run it on the full data set. Run it at the top level as we have done here. And hitting answer live. All right. We will also, yeah, whatever. I'll leave QA and chat open, and we can answer questions in both of those things. Either one is good. All right. And then step 4 of my 4-step process, check the chat in the QA, done. All right, back to PowerPoint. Now I've got too many things open to find the full screen button. All right, here we go. Metadata management. All right, well, this is a short one because this is all I've got written here is, and I've got whatever normalized showing in the background. Yeah, good. All I wanted to say about metadata management, I went right past it, is that it's essentially the data about the data. It's the information that tells you what are these dots. It is the thing that prevents your data from just being a collection of dots and actually informs to what those dots are, what they represent, and how you can interpret them. As you acquire data on your cytometer, you can ask it to record any metadata that you'd like. You can type in and enter stuff. There's a whole bunch of standard stuff that gets added. The standard stuff is generally annotated with a $sign in front of it. If anybody can read finger slashy lines, $sign. I'll show you those in an FCS file. And you can also extend whatever meta information you'd like in Flojo. So let's do that. That was a brief dalliance into slides. Okay, I'm going to close that one. Done with that one, I think. Let's go to, sorry, I've got a lot of different workspaces open. Let us go here. Okay, so I've got some data here in Flojo. I would like to view the metadata. I would like to add to the metadata. I can right-click anywhere on this header and choose Edit Columns. And here is the full list of all the keywords in this workspace. And I can pick any that I'd want to, to show information on. I don't know, we could pick whatever we want. We could do like, I don't know, how about just pick one of the parameters. And that'll appear as a column. And these are generally all editable. So you can, if you did not label what the stain or target was on your parameter, you could do it here in Flojo after the fact. Of some importance is that we never, ever, ever touch your raw data. And so if you hadn't labeled this or you typoed, you could fix it here in the workspace just by typing. Cool. That does not change your actual original FCS file. It's saved in the workspace. If you open and close the workspace, it's still there. If you add the FCS file to a new workspace, it won't be there. You can at any point right-click on a file and export it which makes a copy of that file, and the copy will have any changes or modifications you made to the keywords. You can also do that at the group level. Export / concatenate does for every file in the group. And so if you do change a series of keywords, you can create new FCS files that have those modifications. So that's adding to your displayed list of columns in the workspace, any kind of information that's present. You can also right-click on an individual file, inspect it, and that brings up just this box of here's all the information about the file. That includes on the left all the keywords and their values for that file. It includes if there's a compensation or unmixing matrix built in. It includes all the scaling parameters and a little graph of QC. So nice to visualize the QC. Running one of those algorithms that we talked about a few moments ago is the better way to actually do QC. And then I do want to point out that that $ at the beginning of these keywords indicates that those are keywords that are required to be there by the FCS or flow cytometry standard definition. So any cytometer, doesn't matter who the manufacturer is, will include these keywords. And so you can count on them always being present. There's a handful of them that are very useful. $FIL is the file name. Use that one all the time. $CYT tells you what cytometer this data came from. This, by the way, is just a data set that I grabbed off the Flow repository. I should have cited the author whose name I forget. My apologies. Yeah, we'll put that in the post notes. Anyway, so there's that. $date is the date that it was collected on. Those are pretty useful. There's a ton of other $P or parameter related ones that are generally the less useful once you've already acquired the data. Okay, and then anything that doesn't have a $sign is proprietary. The manufacturer of your instrument decided to include that information or you added it yourself. If you want to add your own keyword, you will go to add a keyword. You can call it whatever you want. I might call it condition. Okay. Oh, sorry. Already have that one, looks like. So I will add a called new condition. There you go. So if it doesn't already exist, you get a blank column. You can just type whatever you want in there. That's fine. Also under workspace and keywords, let me stretch this out to get the full display. You'll notice there's a keyword value series to create a pattern and a copy of value to the entire group. So we could do something here like just associate, how about a number with the condition? And this will be useful when we talk about concatenation later, where we can turn keywords into parameters if they're numeric. So I have got 1, 2, 3, 4 different conditions. So let's create a new keyword that just goes 1, 2, 3, 4. All right, so create keyword value series button. By default, there's a starting value, an increment, and a multiplier. You see we're just going to get 1 to 20 for these 20 samples if we leave everything as default. Cool. I might also, in this case, choose to use a value 5 times before we start over. So you can see I'm getting 1s, 2s, 3s, and 4s. Boom. Okay. We could add a new keyword, maybe call it patient or donor number. And in this case, I've got 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. I might want to, I'm clicking all over, create a keyword value series. I just said create a group from keyword. Sorry. So if you want to create a group from the keyword, you could hit that button. If you want to create a keyword value series, hit the button that, says that. And here I'll just show that you can start over every 1, 2, 3, 4, 5. So we get 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. Boom. Okay. So we've got a variety of numbers added. I should have also mentioned that under create keyword value series, there's another option here for keywords that if you type in some value, every time you find a number, it'll replace it with that value. So if you have something multiple times, it'll put in multiple numbers, multiple iterations of the value. So you can do text as well. I want to do numbers here. As you can see, we've got text, that's fine, but numbers are going to be a little more useful downstream. We also add these as parameters. Okay, I think that is the gist of what I wanted to tell you about keywords. Most of them are written to your file. You can always extend it in FlowJo. Oh, I should mention you can copy paste entire keywords or columns of keywords or multiple columns of keywords right out of like Excel or Google Sheets or something. And also if you're using plate data, there is a tool called the plate editor that allows you to import an entire spreadsheet or CSV file of just keywords if they match up with a well ID. So lots of ways to add keywords. Very important for understanding and working with your data moving forward. Okay. Checking the Q&A box. Looks good. All right. Back to some slides. No, don't blow up the Q&A box. Make it smaller so I can go full screen. Okay, cool. All right, normalization and scaling. Okay, scaling. There is a cytometers preference, so the little heart in the upper right-hand corner flow shown here, and I'll show you in the software itself. And that is going to contain all of the defaults of how we read any kind of data that comes in. We've got defaults for every cytometer. They'll all appear in this list here. If you have added data from that cytometer, we hide anything that you've never added data from before to keep your list specific to you, but they're all already coded. They will generally have a default log, a default linear scale, and whether you want the data to be transformed or not, transform is dependent on the type of cytometer that you add in. We know what kind it is using the $CYT keyword. We'll apply different transformations based on what kind of cytometer. So if it's a mass cytometer, you get the arcsinch function. The other thing that I want to point out is that there are advanced rules that are built into Flojo 1010 moving forward, that if you use those, you can get down to a rule-based method of applying preferences. So here's one, for example, where I did one for the S8. Actually, maybe that's the A8, which is coming soon, which says, I would like a very special set of scales applied to a parameter called eccentricity, because it's an imaging parameter. It's got a weird range. And so I want to say the parameter name contains eccentricity or correlation. I found that those are about actually the same range. And then you can hit the transform button and just use essentially what looks like the T button to make the data look right, hit save. And then as much as as granular as you want, if you want to go to on a per parameter basis, you can do so to set exactly how you want data to be scaled in the future. You could also just say for all fluorescent parameters, do this thing to them. So scaling. Super important, actually. And I'll answer it now, but we're going to also have a user question that touches on this as well. And I'll point to the little bit of text I threw up here on the side of the screen that most algorithms in Flojo are going to do their calculations in graph space. So this is really important, actually. So what that means is that your data has a certain resolution when acquired. You get an intensity value. When we bring it into Flojo, we will bin that data down just a little bit. We will bin it down, some amount for graphics just to make the picture, some amount for statistics. That means small variations in the numbers. So pretty much the data is the data is the data. But when we do graphing, when we do graph space, if we do some compression, if we use something like a width basis or a cofactor for the arcsinch and reduce the amount of visual space that you see given to the negative region, that means the algorithm gets a negative region that has much less variability. And this is generally a good thing. And so we'll get to some question, or at least a question or two where the users were asking about that. But in terms of transform, all you want to do is make sure that you've got all the data on scale, so nothing is smashed on the axes. And for the purpose of doing something like dimensionality reduction or clustering, minimizing the width of the negative population is a really good thing. Mostly we'll think any variation we see just in with background is just fluctuation in noise or measurement error or something of electronic noise, whatever it might be, and not real biology. Those differences, at least, are very hard to ascribe to real biology. And so if we minimize the variability we see in the background, that allows the differences found by the clustering algorithm to be based on your actual biology. And so it's a very nice thing to actually, if you want to get the best possible results, go on a per parameter basis and reduce the variance of the background and to trim out the white space at the edges of your plots. Now, whether you want to keep for parameters of very different ranges of expression, a consistent scale will depend a little bit about your experimental setup. Likely you do. That would allow parameter differences to be maintained relative to how big the difference in their expression versus background is. Cool. But if you just want to have overall the emphasis on the algorithm be whether something bound a particular antibody or not, then you could do on a per parameter basis individual adjustments to the scaling. And so I'll point out that again, we come to the question on that, but that is the gist of this. And like most questions in cytometry, the answer will be a little bit, it depends on what you are trying to study. Okay, so that's scaling. I'll show this in a second, and I see more in the Q&A. We've got one more. Okay. Is there a reference for minimizing the zero negative range? A reference. I think the idea there is that you would just like it to become a fairly narrow peak. I don't think I have a specific number in mind for how wide you'd like it to be. It will vary, of course, based on how much background you have in your sample. Okay, hitting answered on those. Oh, literature reference, I mean. Um, there's probably not a literature reference on that in that the what I've just described is germane to Flojo, where we'll do binning. I mean, it's not germane to Flojo. Like almost every software will do some kind of binning on the data. But if you would take the data to R, then you would have to do your own binning if you wanted to. So there's not going to be an overall um, an overall literature reference, I don't think. Good question. I know how the algorithm will work here, so I can be pretty sure that you will get a nice, less dependent on noise in the background results if you minimize the variance there. Okay. Moving on. Oh, and I always forget that I have that little thing. There is also an auto range built into Flojo. Up to you if you want to use it. That will, on a per experiment basis, automatically set the range for whatever parameters match your conditions there. I've made it not the default so that just in case the first sample that you click on happens to be an unstained control, you don't automatically just set all your ranges to just span background. Okay. Oh, and then here we go. It was the next slide. Here's the user question. So somebody did write in, one can define the same transform for all channels. Yes, you can when you open the T button. I'm sorry, by default, they'll all be the same, unless you do those advanced preferences. And then you can also open the T button and adjust on a per parameter basis if you want. And so it is very important in terms of tuning for the final results, minimizing negative is appropriate. And those tools are real no matter which transform it is you pick, assuming that you've got a transform. If it's linear, for example, there's no compression factor. Evaluating the correctness of the transform. I think most important thing, data is all on scale. Other than that, it's largely aesthetic, and you will get slightly better results if you like in terms of correlating differences in clusters and islands identified in dimensionality reduction if you minimize the background. And the CD3 and 19 for things that have potentially really high dynamic range and strongly bimodalness. Up to you, depending if you want to get results that are dependent on relative expression or just essentially did they bind, did they not expression. Okay. And there was a question on for mass cytometer users, the async transformation with the cofactor. So Flow uses width basis, even when we show the async. The width basis is similar. It is log 10 to the negative of the width basis to get the cofactor. Essentially, you've got a slider and you can make the data squish down the negative population till it looks right, or do, I guess, the math on Excel or whatever to make sure that you've got the negative cofactor matching if you have a specific number you have in mind. Okay. Okay. Normalization. Normalization is the process of attempting to find batch effect. There was difference in the data I collected one day versus the next day, or on this cytometer versus the other cytometer if we're going to compare these two things. This is just a picture that I'm showing that's pulled out of one of the normalization algorithms papers. I don't remember which. The general idea here is showing if you've done something like this is a UMAP of the data, and then I've got all these different isolated populations, and the color scheme indicates what day they were collected on. You can see that the day of collection is actually what's driving the difference, or a large amount of the difference in these populations. Normalize the same data, and all of a sudden you've got islands that are representative of a phenotype that you can compare the amount or the intensity and so forth of, and not have it just come down to what day did I collect the data on. So great in theory. I will caution everybody that if you can avoid normalization, that is probably the best practice by maintaining whatever settings and standards that you can on your machine as best as possible to avoid needing to have it so that background's in the same place and you're working the linear region and all that kind of stuff. And that what normalization's goal is generally to attempt to align data and make sure it looks about the same, which means that if not carefully done, you can use normalization to remove any real result that you might see in the data. That said, there are certainly experiments where if there's batch effect, you may want to try to normalize it out. So a couple of options that you have in Flojo are cytonorm, cycombine, mutual nearest neighbors. I think usually the defining factor in which one of these that you want to use is, did you realize you needed to normalize before you set up the experiment or after? If it's before, then you could use cytonorm, and that would entail you putting in a reference control for each one of the time points or each different cytometer, whatever it is you're trying to normalize against, that you can tell the algorithm that this reference sample should look exactly the same. Figure out what needs to be done to make that so, and then apply whatever you've done to all the rest of the test samples so that we're not trying to normalize test samples to each other. We are saying we've got a known thing that should be the same. Okay? If you haven't done that and you realize after the fact that you need to do some kind of normalization, the other two approaches here are in play. Combine, which actually came over from single cell seq first and was adapted for flow cytometry. It actually essentially uses a regression model to do the adjustment. And I think the neat part of this is that it uses a z-score normalization to, attempt to just say how much different from the mean are you as opposed to actual just raw numbers, which allows for normalization across many domains and so forth. So I'll demo that one. Mutual nearest neighbors is one that's fairly nice in that you can find matching populations, even if there's not 100% overlap of which populations are present. So it's very flexible and extensible. And that uses a correction vector to do its job. So let's take a look at normalization in FlowJo. All right, and I had ran it here, and this took a couple of minutes to run, so I completed it ahead of time. But basically, I just came down to that I had, I imagined it's same data. I had COVID-1 and flu-1, COVID-2 and flu-2, and imagining that these came from, say, one point of collection. Maybe you had one person come in one day and another person come in another day, and you just ran one of each. And so step one on this algorithm is to put some kind of keyword in, just how I showed you could add keywords that identify which batch it came from. So we just did 1 and 2. Next, I had to concatenate. Both mutual nearest neighbors and Combine will run on one file and produce a combined normalized set of parameters. And so we'll put them together via concatenation. So this is done by selecting the population of interest. Both of these algorithms suggest that you do a little bit of cleanup, just like we talked about at the beginning, because they'll both use a clustering step to try to identify large populations in there so that when we're trying to normalize a given parameter, we're not expecting, say, the CD3 distribution to be exactly the same on a population that's largely made up of T cells and another one that's largely made up of dendritic cells, something like that. So concatenating could mean highlighting each of the populations that you want to move forward with. It could also mean right-clicking on one of these and saying select equivalent nodes. It'll highlight that population on all the files. Here, I've actually already got the concatenated file in there, so I might want to just pick the specific ones I want. I right-click. I say export/concatenate. Choose the concatenate option. You also might want to do this on the compensated parameters, so all comped might be a good thing. If the data have inherent compensation stored as a keyword within the data file, meaning you compensated or unmixed on the cytometer and exported it with the matrix, embed it in the file, then either one of these choices will effectively be the same. And under advanced options, choose whatever keyword you used to link together those files. So I called it batch ID, pick that, say OK. When we add a parameter, add a keyword as a parameter, we will spread the distribution by default, just add some noise so that you can see it. But for this purpose, we want to auto-gate and figure out which batch these are. Get rid of that noise. Just say, I just want basically a straight line for all the cells in one file and all the cells for another. You'll say concatenate. Oops, let me give it a different name to not to not overwrite the one I already have done. XYZ. It'll write the file. There it is on my desktop. And then you can either click existing workspace to bring it right back into this workspace, new workspace to put it in a new workspace, or just close and that file's sitting there and you can drag it and drop it into any workspace you want. So I had the concatenated file here and I can go in. These are all lymphocytes. You'll now see, even though I dropped it into a group with lymphs, there's 100% in that. We can say workspace, plugins, and Combine. Okay, select the parameters to normalize. Generally, it's going to be all of them. That is what, sorry, all the fluorescent ones. That is going to be what is highlighted by default at the top. You can also, and it's going to make a new file called normalized concat, you can view and edit which parameters are going to be written into that new file, because these things may not be the same. By default, everything is written into there, including things like forward scatter and sample ID and time and all the things that you probably don't want to normalize. But you can have them export it or remove some if you want. All right. It's going to use a self-organizing map to try to identify the major populations in the data. It gives you a default grid of 8 by 8 or 64 nodes, so 64 different possible populations can be identified. That's probably plenty. Z-score normalization versus just a rank score. Z-score is like difference from the mean. Rank is just order of these things being different. You've got a cofactor for transforming the data, and you have to identify which one of the keywords is the thing that is going to tell you which batch it is. You hit OK. This took 4 or 5 minutes to run. We won't sit here for 4 or 5 minutes on a webinar, and it produces a normalized underscore, same name of the file with the method used. And then if I go under here, then each of these comp parameters is now a normalized version of what I was working with before. Plus, if I go to the layout editor, it actually gives me a plot showing before and after for each one of my parameters, before and after normalization, that is. So you have a feel for how much has happened. So I just ran it on this data file. There probably was not actually a batch effect on that, so there's very, very, very minor corrections. You can see barely anything, almost complete overlap. But the gist of how you run this would be exactly the same, even if there were more major corrections. Okay. All of the different tools that I presented in the slides will work about the same, where you'll pick a population, you'll go to plugins, and you'll pick what you want to run. So MNN is there. The CytoNorm one is the one that is a little bit unusual in that it is a workspace plugin. And so you would actually have to go to the Add Workspace plugin, pick CytoNorm from here. The gist of that is that node-based plugins run on something already in your workspace. Workspace plugins pull something in essentially at the add or save moment of a workspace. You would point it at a folder, and it would pull all the things in a folder and do the normalization at that point. So works a little bit differently, but same basic process. Okay, now let's go look at the chat. IIRC CytoNorm recently proposes a mode without standard sample in each batch. Yes, this is true. The statement that I made there about, so you have to have thought about this ahead of time and prepared a standard sample, made the group that does CytoNorm think, well, maybe we could do one without. And so they've attempted to have one that works that way as well. Okay, Anonymous says, dun, 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 dun. What is the best way to identify if normalization is required simply by viewing multiple batches to notice if there are any gross shifts? That is a way to do it. The other way that was shown in the slide, there was a lot of folks who use some kind of dimensionality reduction tool and see if an overlay versus sample produces clusters that are specific to the time point, as opposed to clusters that are comprised of similar events from multiple time points. Okay. Cool. Back to PowerPoint. Okay. You know what? Having had to concatenate to do the normalized bit, that makes this concatenate section slightly less useful, probably. I'll do a little bit more of an intense calculation here. But anyway, concatenating, putting things together. The overall general utility of concatenate is making sure that you've consistently applied some kind of algorithm. So whether it be dimensionality reduction, clustering, whatever, a lot of these algorithms are stochastic. They've got some kind of random aspect to how they're initiated. There are ways to use seeds to de-randomize and so forth. But there's some really nice utility not worrying about anything about that and just running it on all the data at one time, so that if you've got cluster one, it's the same cluster one in all the samples. If you've got a group of cells at the top left of your t-SNE plot, it's the same phenotype in all of your samples, as opposed to, you'll get pretty consistent results on all these things, but some of the metadata might vary. Like the B cells might go to the top in one run of t-SNE and to the bottom in another. We can avoid that by doing concatenation. I will talk a little bit later about how do I extend any kind of result. That is something a user wrote in, and we have an answer for that one. But I want to mention also here that it pairs nice with keyword labeling, so that you can extract the initial information of these were different samples with perhaps different conditions and so forth at any point. Okay. question that was written in. What are best practices for concatenation when you have a large number of samples, e.g., 8 time points times 20 patients? Is it best to include an equal number of events from all 160 samples in the concatenated file? Are there guidelines for number of events to retain from each sample when downsampling? So, good question. The answer to this is that, besides it depends, you are going to use the concatenated file to essentially make a model of what your data looks like. And so what you'd like to make sure is that the model can represent all of the different conditions that are represented within all of your data files. So if you've got a really rare population that you want to make sure is included, you may not want to downsample. Or you may want to do the most minimal downsampling that, if you check the statistics, means that you're going to get some of those cells at an appropriate number in it. You can check based on what the expected frequency rate of that population is. I also don't think you necessarily need to have exactly the same amount of events in your representative files. If you represent a condition fairly equally, that's pretty good, and fairly is close enough in that it's cytometry. We will be talking about tens or hundreds of thousands of cells, and even if we get down to some smaller populations, we might be talking about thousands of cells. And so if you've got 50,000 of one kind of cell from one type of patient and 54,000 from another, though that's not exactly the same, there's plenty of data there for you to be able to model that particular condition. So you want to make it close. If it's off by an order of magnitude, that means you're largely going to be modeling one condition and not the other, so that might not be optimal. So I think the answer there is it doesn't have to be exact. There are tools in Flowjet to make it exact. Try to make sure whatever your target population is going to be included, and try to make it as even as possible while keeping as much data as possible. I think that's the big thing for me. You want to keep as much data as possible. The algorithms at this point will run fairly quickly, even on large data sets, or if you've bothered to collect all of this data, and it's tons of data, it might be worth letting them run an hour to get the maximum impact of all of your data. So let's talk about exactly how to do all of that stuff. I'm going to go back to, I think we're done with this workspace now. I'm going to go back to this one where, oops, sorry, wrong one, where that is a preview of what's coming next, where we've added a condition and a donor number for all of these samples. I think one thing I didn't have explicitly noted there when we do cleanup gates and so forth is you also might want to do a phenotype gate or two before you do any kind of clustering or any kind of dimensionality reduction. If your experiment is, as it was in this case, to do something like identify all the T cells, and you're not going to look at anything, or like different flavors of T cells and whatnot between these different conditions, and you're not going to look at anything that's CD3 negative, then if you feed a CD3 negative population into your t-SNE algorithm, your UMAP algorithm, your flow sum clustering, it's going to find major differences between the CD3 negatives and the CD3 positives that will dominate the results, and you might not care about that particular difference. And so it's probably worth putting in a CD3 positive gate and isolating what is done on your data to the stuff you care the most about. It's also possible to do these things recursively, to run a clustering algorithm or whatever to identify the CD3 positive population within that automatically and non-heuristically, and then run it again on just those cells to find out what the different flavors are. Your call, but there's probably a thresholding step where if you want to really get into the nitty-gritty of a particular subset, examining that subset. So just for funsies here, let's pick, I don't know, this is a full data file. I will just say something like CD3, just to illustrate that we could make a gate if we actually bothered to make a gate. Yeah, whatever, I'll just pick CD3 and side scatter low because that's what I happen to grab. As this is just for illustration that I could pick a population when I do concatenation, I kind of showed that before. We'll do edit, select equivalent nodes, boom, grabbed all the CD3s here. I'm going to then right click on this and say export/concatenate, concatenate. And now I've got new condition and donor number that I want to add and convert from keywords to parameters. And so I'm going to find them in the list. It's alphabetical. So condition and donor number, boom. Actually, I want a new condition. Sorry, condition was a bit of text. Numbers can be graphed. Text can't at the moment. So keep that in mind. Okay, boom. Got two keywords. We'll spread the distributions here so it looks a little easier, nicer to see. Call the population whatever you want. CD3-ish. Okay, again, all uncomped gets you the uncomped data. If you have a keyword compensation matrix that's the same for all of these files, which is true for this, then it's the best of both worlds. I get the raw data and an appropriate matrix for all of them. If I've brought these in from different experiments and they each have their own compensation, or I've compensated in FlowJo, you'd want to pick the all compensated parameters that we apply compensation or on mixing on a per cell basis. So you get the appropriate one on that and then can do the comparison afterwards. Cool. All right, hitting concatenate, this box pops up just as you saw before. It's making this concat CD3 file that will appear somewhere on my desktop when I write the megabytes. Here I'm going to say existing workspace and hit close. Okay, and now I've got a 49th sample that, if I go to all samples, will be somewhere here right there. And the thing that I want to point out to you now is that, so I've got this file, I could run my tsne on it, I could cluster it with whatever I want to cluster on, or I could UMAP, whatever I want to do. But I can also go in here and I've got donor number, I've got new condition, and so I can get back to each of the initial files if I wanted to. A very fast way, actually, by the way, to isolate by type is if you make it a histogram, you can right click, you can say create gates on peaks. We will just name these 0, 1, 2, 3, 4, 5. But for example, these are the donors. So actually, I guess that would just be pretty straightforward. If we had done that by new condition and right clicked and said create gates on peaks, then if I want it, I could go down here and actually just right click and rename and say, yeah, well, I know the first ones were COVID and the second ones were healthy and so on and so forth. So you could actually break apart these things by patient, by condition. If you want to break them apart by both, you can drag and drop either way. So there's the COVIDs broken down by patient number and so forth. So you can get back into an individual file or gate on everything by condition or everything by donor or whatever other keyword you've added in there so that you can do comparisons, post-clustering, post-dimensionality reduction, and so forth. Okay. I should point out that if you don't add any new keywords, FlowJo does add the sample ID keyword, which just breaks down each individual file into just a sample number that fits in the standard 0 to 262.144 scale. So you can get that out. Question from the audience here. Did I hear you say that spectral unmixed data should be concatenated using only compensated data? Can you repeat the guidelines for making the choice? The biggest difference there is whether you have a keyword that includes the matrix. So if that keyword is uniform for all the files, and regardless of whether it was unmixing or traditional compensation, you'll have everything in there. And if you've got already a spectral mixed data file that came off, say, well, any one of the cytometers that do spectral and mixing, it may be embedded in there already. So if the keyword value contains the mixing, you're good to go. If you're bringing in data that have different unmixings or compensations and want to concatenate them into one file, the way to preserve the unmixed value for that individual cell is to choose the compensated data choice. Okay, so the biggest difference there is not whether it's spectral unmixed or compensated, it's whether all of the files have the same thing or not. And also, I guess, if it was done in FlowJo, then there isn't a keyword that lists what your unmixing matrix or your spillover matrix is. And so you'd want to do the compensated parameter checkbox there as well. All right, checking. Questions 2. Does FlowJo have a max number of events that can be concatenated into a single file? It is not a max number of events. There is a maximum file size that we can handle that is a matrix of the number of events multiplied the number of parameters. It's really big. Generally, the limit happens on if you get to files that are approaching that limit, it starts running slow. It's like, God, I don't remember offhand. I think it's like 8 gigs or something like that, I think. But it's pretty big, but it's not, there's no specific number of either events or parameters, but the combination of the two, which lead to a larger file size. So there is some benefit if you care to of when you choose which parameters to export into a concatenated file of leaving things out perhaps that are no longer useful within the realm of possibility, for example, that you take your live dead die and you gate on the live things and you're like, I'm not really going to use the dead cells for anything anymore, and just leave the live dead marker out of subsequent exports. FlowJo will run faster on smaller files. There's just how much data you're lugging around bit to bit that actually makes a little bit of a difference. So up to you. Okay, so I think that was your question and like two others that I just shoehorned into one. All right, checking the Q&A again. All right, we're looking good. We have 5 minutes left. How are we doing? We might run slightly over, but when you've got run-of-the-mouth disease like I do, sometimes that happens. Oh, we are on to do great things. Sweet. We may actually land this plane. Okay. Uh, Question from somebody who wrote in. If additional data from the same project panel become available over time, e.g. new time points or new patients are added, is it best to start the workflow over from start with representation of each new plus old sample in a concatenated file? Great question. So a lot of people ran into that exact workflow problem where, yeah, I've got this, T-SNE plot, and this particular island is really interesting phenotypically, but now I just got a new data file, and I don't, how do I know if that population exists? You could, because of the stochastic nature of the process, start over to make sure that you just have a new data set, you identify the new interesting population, or you could use generalized embedding. So there's an extra algorithm that we have put into Flojo. The general gist of this is if you have done the steps we described previously and then came up with clustering results or t-SNE results or something like that, and then get a new data file, you can use the old data and the original measured parameters, so the CD3s, the CD19s, etc., to say for the new file, find me the nearest neighbors, the cells in the old file that most resemble this new cell from this new file. And this example has 3 nearest neighbors. We actually use a default of 7. You can adjust it if you want. But we also weight it for, are you the nearest? Are you the second nearest? Are you the third nearest? And we say, okay, so here's our weighted score of these are the 3 most similar events. And then we say, well, what were their t-SNE values? Or what was their cluster number? And if it's something like t-SNE or UMAP, then we take a weighted average and create t-SNE or UMAP values so that a T cell falls onto your t-SNE plot in the same place as the rest of the T cells. And you can directly compare these plots to each other. If it's a clustering result, we'll actually just vote at that point and say, okay, well, you were cluster 1, you were cluster 1, you were cluster 1, you were cluster 2. We'll put this cell in cluster 1. That way you can directly compare this by just forward propagating basically the results done previously to the new file or files. And so that takes a lot of burden off you as the experimenter to put everything together or have to start over the process, which sounds ugh. Anyway, let's look at this tool. I did notice a QA question popped up. Boom, boom. Do you have recommendations on computer RAM and processors to run the plugins on larger data sets? The general recommendation for the RAM is that the hardest part of RAM is if you've got a data set that's bigger than the RAM and you have to then offload and reload bits of your data set. If you can see how big your data set is and get your RAM to be large enough to encompass it, that's the best case scenario. So it'll vary based on what large data set means to you. All right, so let's take a look at this tool. There's the thank you, because I can't hit the escape button without accidentally advancing, apparently. Let me go back to this workspace here where I set this up. Okay, so I concatenated a COVID sample and a healthy sample, and I ran an algorithm called T-Rex. It's one that both does clustering and dimensionality reduction. It attempts to find out the most significant clusters automatically, and I came up with t-SNEs and T-Rex clustering scores for this concatenated file. But then I got a new file, and I said, what will I do with this new file? Let's embed so that we can do direct comparison. Okay, so I clicked on the population that I had used to like do the original T-Rex on, and I go to plugins, and I go to, sorry, not a plugin. I go just the embed button. I hit the embed button, and up at the top is what's my target. So this is where you'll pick out your t-SNE parameters, whatever dimensionality reduction you're using, whether it's UMAP, etc., whatever your clusters are. So here I would say t-SNE, t-SNE. I will want to get the clusters but I also want to click over here for type and make this a cluster parameter. For a lot of our clustering algorithms, embed will just automatically recognize that, oh, you're FlowSum, I know what you are, and will know it's a clustering algorithm. If you throw it something like T-Rex, which is relatively new and we don't immediately identify it, right click and choose cluster. The bottom is, so which parameters are fair game to try to ascertain phenotype to match these up? And it's generally going to be all of them. So I would pick them all. Boom. Okay. The rest of this is probably fine to use the defaults. The only one that you might want to think about is right here. There is a 4 cells to be placed in a cluster. And with that one, you can imagine a scenario where we've got 7 nearest neighbors and a cell whose nearest neighbors are in cluster 1, cluster 1, cluster 2, cluster 2, cluster 3, cluster 3, cluster 4. Who wins the vote there? I don't know either. So we would just put that event into cluster -1, so it stands out on the side as didn't fit into anything. Or you could check that box and we'll just put it into one of the clusters, generally whichever one came first, if it has to be forced to be in a cluster, even if there's no clear winner. I hit accept on this. So that'll think for just like a second or two. And then that's going to pop up a message that says, ready to drag. At this point, you've got your trained model, drag it and drop it to anything that you want to apply it to. So for example, I could come up here and I could just drag this to, I will mention these were actually trained on CD8 T cells, so I could drop it on the CD8 T cell population. This took me about 4 minutes to run it on COVID-2, so we will fast forward that. We'll let COVID-4 think for a little bit and say the checkbox appears once it completes, and then you will see the embed version of all those parameters pop up. You will also get, as long as you left the default checkboxes on input quality and output quality. So if I go to this population, oh yeah, I did that too. So if you go to this population, so here is just a plot of what what the results look like using the whole t-SNE parameters there. But let's start with input quality and output quality. You still here? I think we're still here. Sorry, there's a storm brewing here and my internet just froze for a couple seconds. Let's see. We still have 11 participants. I think I'm still talking. I think you can still hear me. Maybe. Oh my. I'm gonna keep going in case you can hear me. Zoom. Flojo's working, but Zoom looks frozen, I think. All right. So anyway, I'm going to go to input quality and output quality. This is the last thing I'm showing anyway. Hopefully we do okay. All right. OQ and IQ at the bottom of this list here. So what, so let's not show the -1 since it doesn't really look like anything was in the -1. Let's customize this axis down to just start at 0. Okay. Oh, all good. 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 All right. Oops. I did something where I want to customize that and start at 0, not whatever happened there. Okay, cool. All right. So it's going to find the near neighbors, whether they're actually near or not. So input quality and output quality. Input is, are my nearest neighbors actually near? And let's end this at about 1, because that's what the scale goes to. And we can bring this to like 4. Cool. All right. So for the cells with the smallest values, both the input neighbors were very similar, and the t-SNE results were very close to the nearest neighbor's t-SNE results. They're the ones that will map perfectly. The ones that are farther out, there wasn't a great example in the original data. And so maybe those are the most interesting cells that a novel population occur, or ones that you might want to ignore. So if you want, you can also like throw a gate on here and choose by sample quality which ones you want to look at. And again, it might be the best or the worst ones, whatever. Double click on that and you've now got let's just go there. TSNE1, TSNE2. You'll have the clusters as well. What you would want to do with those clusters is to put them on a histogram and AutoGate because it'll just add the parameter there. Okay, so that is the COVID-2 sample. And so let's go back to the concatenated COVID versus healthy sample where we had COVID and healthy. And I just showed them on the same plot. And so here's kind of what you're hoping to see. So I've got many, many more cells in this concatenate one that I made with lots and lots of samples. Big, huge fat population that's present in both the healthy people and the people with COVID. So here's COVID, here's healthy. In fact, a lot of these populations look like they're fairly equally represented. Maybe there's more cells here in healthy than here in COVID, but a big population emerges here that happens to be Ki67 positive. That's kind of absent in healthy folks. So we apply this t-SNE to this COVID sample. And if we hadn't known what kind of sample it is, we say, oh, well, look at that. There's an awful lot of cells down here in this population that are Ki67 positive that weren't present in the healthy folks. I think this might be a sample that is from somebody with COVID. And so it's a way to forward propagate any of those results without having to rerun everything on all of the samples again and to get a consistent population that you can look for if it has any kind of discriminatory power. Okay, looking at the QC or the QA, the QA again to see if there's questions. We are 6 minutes over, so already I appreciate you who have stuck with me for an extra time, even through perhaps freezing on Zoom. Woo! We can also see if COVID-4 finished. Yay! So that one's done. We could look at that too if we had time. Looks like we are good in the QA. Let me just check the chat to see how that's doing. We've got 2 chat questions. Okay, people who were telling me they heard me while Zoom was frozen and I couldn't open up the chat, so thank you for that. Okay, thank you for joining. Last chance to ask a few questions, and if not, we will have this one posted to the recording session in an hour or 2, I guess, or whatever it may be. And other than that, thank you. Enjoy your upcoming holidays. Thank you, Samuel. And turning